next talk is another rapid-fire talk. Number 18, circulating markers of matrix homeostasis are dysregulated in hypertension-induced aortic stiffness, presented by Dr. Gedney, senior author Dr. Ruddy from the MUSC group.
I'd like to thank everyone for the opportunity to present here today on our recent work in investigating circulating markers of matrix homeostasis and their dysregulation in hypertension-induced aortic stiffness. I have no disclosures. Aortic stiffness is a major predictor of cardiovascular morbidity and mortality, and aortic stiffness is associated with hypertension. This is thought to be due to changes in vascular smooth muscle cell signaling, as well as inflammatory infiltrate and proteolytic remodeling of the extracellular matrix. Intrinsic stiffening of the aortic wall structural components can propagate dysfunctional signaling of signature remodeling peptides that persists even after normotension is achieved. So clinically, the assessment and evaluation of aortic stiffness has not been widely adopted to inform medical management, and furthermore, correlation to circulating biomarkers are poorly understood. So we hypothesize that plasma markers related to matrix homeostasis are associated with increased aortic stiffness. Wild-type mice had angiotensin II-induced hypertension for 21 days. Systolic blood pressure as well as ultrasound-derived strain parameters in plasma was collected. To assess the aortic wall mechanics, a myograph system assessed induced tension at experimentally derived optimal tension as well as optimal tension plus 30% for 6 hours. A subset of these was also exposed to angiotensin II. To assess vascular smooth muscle cell response to mechanical stimulation, we induced 12% biaxial cyclic stretch for 6 hours. In each of these cases, the expression of aortopathy-related proteolytic matrix markers was evaluated. So at 21 days, we saw a 30% increase in the systolic blood pressure of our model. We did not see a change in strain of the aorta, but we did see significant changes in distensibility and pulse propagation velocity that demonstrated an increase in aortic stiffness of our angiotensin II-induced hypertensive model. In the plasma of hypertensive mice, we saw a statistical increase in the expression of IL-6, which is an inflammatory mediator of macrophage expression. The gelatinase MMP9 decreased in its expression. The serine protease antagonist system of both cathepsin S and cystatin C both increased with hypertension. We saw no change in OPG, which is a vascular marker of vascular calcification. And then tenascin C, which is a transcriptionally active matrix protein, we saw nearly a 3-fold increase with hypertension in plasma. Our proteolytic markers were also assessed in our myograph system. The myograph system was meant to exclude inflammatory infiltrate that we see in our hypertensive model. So we expected to see a differential expression in our markers, but we saw something similar to our in vivo model. IL-6 increased, cathepsin S increased, cystatin C increased. OPG did not change, and tenascin C increased. And then drilling down into our vascular smooth muscle cells response to mechanical activation, we saw an increase in IL-6, did not see a change in MMP9 expression. Cathepsin S increased. Cystatin C did not statistically change. OPG did decrease with mechanical activation, and then tenascin C increased. So in summary, the increased aortic stiffness and angiotensin II-induced hypertension can directly be measured in the abdominal aorta by ultrasound. Plasma levels of inflammatory cytokines, proteases, and fibrillar proteins increase with aortic stiffness. And in pathologic remodeling, the expression of aortopathy-related proteolytic matrix markers is influenced by biologic and mechanical stimuli. So in conclusion, tracking aortic stiffness and proteolytic matrix markers may direct medical therapy and hypertension to reduce cardiovascular morbidity and mortality. I'd like to acknowledge and thank the members of MUSC's Cardiovascular Research Lab for their help with this project, as well as my funding sources. Thank you.
So I'll ask a question. So how close are we, time-wise, to having this be a targeted therapy? This information lead to a target?
So this study is purely descriptive of what is going on in the aortic wall. We can't specifically say that we can track one of these yet, so I think it's a long way off, but it is interesting that we're able to, descriptively describe what's going on in the aortic wall and how that's expressed in plasma, which is just one step closer to being able to track something in a patient's blood over time.
So is the thought that somehow this inflammatory response induced by that begins maybe in the aorta, then downstream consequences as far as other vascular beds, atherosclerosis, potentially aneurysm development?
Yes. So the idea is that this study focused on hypertension and how that affects vascular remodeling, but you can extrapolate that to other areas of vascular disease, including aneurysm, peripheral arterial disease, that hopefully, that's something in the future to study, but we focused on hypertension right now for early remodeling.
All right, nicely done. Thank you.